In this video, we will discuss about how the CD44 on NK cells could be the potential mediator to CAF, that is cancer-associated fibroblast, and malignant epithelial cells. So, in the panel A, what they did is they did the cell chat software to identify the interactions of between this epithelial cell, different kind of epithelial cells, that is epithelial cells that are expressed in the or malignant epithelial cells and normal epithelial cells, fibroblast and NK cells. And what they have been able to find out is that the outgoing interaction strength of this fibroblast is the highest, and also this epithelial cancer cells ingoing strength is the highest. However, the normal epithelial cells does not have any difference in the or any significant difference in the incoming and outgoing strength, and the NK cells also has this incoming strength, incoming interaction strength. So all this indicates that and these epithelial cells also has this epithelial cell fibroblast also has some incoming strengths, and this triangle and circle represents the different replicates. So all this shows that apart from this epithelial normal cells, all other has some increased in the interactions, whenever the pancreatic cells converted into the it's converted into the cancer cells, and they have oh, this is not replicates. The triangle and circles are not replicates, but this triangle represents the adjacent normal cells and circle represents the uh, cells that has this PDAC disease. So we can see that there is not much change in the epithelial normal cells and epithelial normal cells between the PDAC and adjacent normal, but there is a significant change of we can see in the fibroblast between the PDAC characteristics and the normal characteristics. And same things we have seen for these NK cells and also for the epithelial cancer cells. So this indicates that there is significant changes occurs whenever the normal cells converted into the cancer cells, and when they wanted to check the pathways that are also get dysregulated, they are able to see that some of the pathways get dysregulated, and mainly are this collagen, laminin and FN1. These are the some of the pathways that are highly dysregulated compared to its adjacent normal behavior. And now they wanted to see the ligand receptor interactions and with respect to the NK cells, that how these signaling pathways are affected by the NK cells, and they are able to find out that it's the CD44 that plays on the NK cells that plays a major role towards triggering this change in the signaling pathways. For example, this epithelial to cancer cells and NK cells interactions mainly cause on this CD44 and call 9A3 interactions and the other interactions that are shown here, and the color represents its intensity. And so maximum intensity are be found here where the fibroblast and NK cells interactions occurs, uh, that is CD44 and call 1A1 and CD44 call A2 call 1A2. These are the places where maximum interactions occurs. So this all indicates give them the hypothesis that with increase in the collagen, laminin and fibronectins, the NK cells might be get trapped by the tumor microenvironment, and there it uses overexpress this CD44 to interact with the tumor microenvironment and trigger the signaling change in the signaling pathways. So now they wanted to check whether the this is their hypothesis is correct or not. The good way to check is that to see the CD44 expressions in their malignant versions or in the malignant cells. So first they check the CD44 expressions in the normal cells. First they check the epithelial normal cells. In the epithelial normal cells, the CD44 interactions. In the adjacent normal cells, they do not, and the PDAC cells, they do not see too much changes in the CD44 interactions for the epithelial normal cells, but when they check it for the epithelial cancer cells, they can see the increase in the CD44, and this increase is significant compared to the adjacent normals epithelial cells CD44 interactions. And when they check it for the fibroblast, they do not see the increase is significant. CD44 increase in the PDAC fibroblast and the adjacent normal fibroblasts are significant. They do not see any significant increase of the CD44 signals. But for the NK cells, they can see the significant increase. That is, for in the PDAC regions, the NK cells has this higher CD44 expressions compared to its adjacent normals NK cells, which is a normal healthy NK cells. So this all indicates that the CD44 expressions are higher in the case of these NK cells when are when in the PDAC conditions or are surrounded by this fibroblast. And same things they have seen for these epithelial cancer regions, where we can see that the epithelial cells acquire the cancer properties or malignant properties. They shows the high expression of CD44 because epithelial cells, from our previous videos we have understood that epithelial cells and cancer NK cells are found in the same region during this tumor microenvironment development because of the pancreatic ductal adenocarcinoma. So these results further substantiate their previous finding also, and they also provided us a new insight that CD44 on the NK cells get overexpressed in the tumor microenvironment to deal with their malignant behavior within the tumor microenvironments. That's all for this video. So if you need to understand more about this experimental results in detail, you can go through the reference that has been provided in the description. Thank you for your time.